My guest today is Professor David Pearce, who's Professor of Microbiology in the Department of Applied Sciences at Northumbria University. The underlying theme of his research is to use microbiology to understand polar ecosystem function and the potential for shifts in biogeochemical activity that may result from environmental change. Welcome, David.
Thank you very much. Glad to be here.
Yeah, so this is a bit unusual for me, David. You're talking to me from a ship in the Arctic,
Yes.
and our connection appears to be pretty good. So the internet must be working. Um, and so you send me your papers. I want to go through them. They're all very, very interesting to me. I know nothing about them,
Okay.
but the topics are quite interesting. So I want to start with functional associations and resilience in microbial communities. So you say here microbial communities have inherently high levels of metabolic flexibility and functional redundancy, yet the structure of microbial communities can change rapidly with environmental perturbation. To understand whether such changes observed at the taxonomic level translate into differences at the functional level, we analyze the structure of taxonomic and functional gene distribution across Arctic and Antarctic locations. So let's get some definitions. So taxonomic, my naive thing will be this is what humans put them into categories, right?
Absolutely. So, first and foremost, I ought to mention the polar angle. One of the reasons why I, in particular, study polar ecosystems is because originally we thought they were relatively simple systems with low biomass and low taxonomic diversity, so small numbers of different species that were easier to understand and easier to put into context and easier to model. Now, that's not necessarily the case. There are biodiversity hotspots and biodiversity not spots, but it is because it's a stressful environment for the growth and reproduction of organisms, it is easier to constrain what they are and what they're doing. So this paper itself, historically, microbiologists have looked at microorganisms by trying to name them and categorize them, much as Victorians used to do with higher plants and animals. The first thing we have to do is name what we're studying and understand how they differ one from another. And with bacteria and viruses, it's very hard to do that morphologically. You can't just look at them and say that's the shape of a tree or that's the shape of a fish. They all tend to look very similar. So the way we differentiate bacteria in particular is to use a gene, a highly conserved gene called the 16S ribosomal RNA gene, which is highly conserved across all different organisms. So there's a database of these genes, and we use that as a benchmark for taxonomy. So instead of saying there's John, Peter, Paul, for example, in a community, we might say there's a bacillus, an Escherichia, a Shigella, what have you. But what does that actually mean? If I use the analogy of a human community, if I'm in my work environment, there might be 1,000 of us at the university, one of whom speaks Russian, just a random language taken at random. But that's a useful function for the university. One of the members of staff speaks Russian. Now, if I transfer that analogy into the bacteria, there might be 1,000 species of bacteria, one of which can fix nitrogen. And therefore, that nitrogen fixation function becomes very important. And it doesn't matter if it's an Escherichia, a Shigella, or what have you, a John, a Peter, whatever. The fact that that function is owned by one bacterium is what really matters for environmental function. And yet, all the time, as microbiologists, we're concerned with naming them, giving them a taxonomic affiliation, or calling them something. So I think what this paper really is about is getting away from the names, trying to understand what's important about what microbes do in terms of ecosystem service and how resilient they are. So if we take one of those species away, will another one do that function instead? Or does the ecosystem lose the ability to have that function? And that's where the resilience comes in. And what that particular paper shows is that communities are quite resilient, and it doesn't look like they're vulnerable at the moment, but theoretically, there is a point at which they could collapse. And we may be a long way from that at the moment, but technologically, we're just at the point where we can understand that that collapse is possible, and this might be why.
Yeah, this is really interesting, David. So I quite like this functional angle.
Mhm.
Um, so the functions are more scientific than taxonomic
Mm,
because it's somewhat arbitrary in many ways
yeah.
based on the current understanding, right? So you say here we found the functional genes distributed evenly
Mm,
across bacterial networks,
yeah.
and that this functional distribution was also even across different geographic locations. That's
Yeah.
surprising, isn't it? I mean,
It's incredibly
yeah.
powerful. I mean, that I found it, I struggled to get this paper published, not because there's anything inherently wrong with the idea or the hypothesis behind it, but because I found it very challenging to get other scientists to see the importance of the observation. And I
Mm,
think a lot of the journals were a bit concerned about had they published it,
mm,
they could be taken into ridicule or question or what have you. And it's a bit like Charles Darwin must have felt when he first came across the theory of evolution. He had an idea. That's all it
yeah.
was. It was an observation. It fit the observed data that he had. And then he went out and said, I think this is a fact. And everyone said, no, that's rubbish. That's the stage at which this particular paper was quite
Mm,
groundbreaking in the sense that it shows for the first time that data upholds this idea that these functional associations exist, and it
yeah.
doesn't matter whether you have the list of organisms in the environment, and
Yeah.
that environments can be very resilient in that if we use
Mm,
the nitrogen fixation
mm,
analogy, if one species doesn't do it, then another one will. So you have this what we call functional redundancy. So you
mm,
might have 10 species that can all fix nitrogen, but only one of them does it routinely,
yeah.
unless that's removed from the system, and then one of the other nine will come to take its place.
Mm,
And because that happens, the environment never loses the capability to fix nitrogen. And so we
mm,
say it's resilient. And
yeah.
from an environmental functional perspective, we all worry as humanity that environments can lose their functionality or that things can happen. Climate might change. We can
Mm,
pollute waters. We can affect the distribution of organisms in a way where you actually lose function. And that's a serious worry. But now we're starting to see, well, they do look to be quite resilient, but
mm,
there is a limit to that resilience. And so the challenge now will be properly defining that level of resilience, working
mm.
out where the limits are, and how much flexibility we have in the system to challenge it in a way where it can still get back to its original function.
Yeah, so it's a bit counterintuitive for naive people like me. Um, so we have been sort of fretting over are we losing diversity? But in the microbial world, there is redundancy, and that gives us resilience. So you say here, for example, on average, 15% of the functional genes were related to carbon cycling across all bacterial networks. 21% of the genes were stress related, and only 0.5% of the genes were linked to carbon degradation functions. Um, in some sense, this is sort of evolution, isn't it? I mean, when there is a gap,
Mm.
uh, biological entities step in and
Yeah.
do things. Yeah.
So it's one of the reasons I'm a microbiologist. I started my career as an ecologist and I work with whole animal systems, but it's incredibly difficult to do experiments on a meaningful scale with higher animals and higher organisms simply in terms of the population numbers and the times it takes for the populations to turn over. But bacteria you can grow in there tens of thousands, hundreds of thousands in a 24-hour period, and you
Yeah.
can do it. You can do experiments that you wouldn't even contemplate with higher animals. So it gives us a much deeper insight into ecological theory. So you talked about biodiversity loss, and in the world around us, we think of the challenge we have at the moment, one of the core challenges for humanity is biodiversity loss. And we are
Mm,
seeing loss of ecosystem services, ecosystem function, and we are seeing the reduction in the number of species present. But
mm,
what does that mean in terms of what are the consequences of that? Because
mm,
we are currently in the sixth mass extinction.
mm.
So if you look through evolutionary history, you see these pinch points when the number of species overall gets reduced, but we
Yeah.
seem to emerge from that and the planet recovers like with the loss of the dinosaurs. The plant does recover and it repopulates, and those ecosystem services are reinstated, albeit by other species.
Yeah.
The current mass extinction is worrying because it might include ourselves, but let's not get too worried about that at this point. What we can do with microorganisms is understand how this resilience pattern works, how this functionality works, how it can repopulate, and how it can resurge after a bottleneck.
Mm.
Like that.
So, talk a little bit about sort of the functional gene system.
Yeah, sure.
So
Okay.
how are you determining this? This is what sort of experiments do you do? Yeah.
Sure. Well, there are many different functions in the environment that microbes undertake. The obvious one are biogeochemical cycling. So what most microorganisms are known for, including pathogenic ones, is breaking down and recycling organic material. So if a tree dies, fungi will colonize it, they'll break it down. All those nutrients will be recycled into the soil and a new tree can grow. So the primary role of microbes is breaking down and recycling organic material. But that can also be applied to all of the core nutrients, things like nitrogen, phosphorus, sulfur. Microbes can move them through the environment. They can also fix atmospheric gaseous nitrogen so it can be turned into proteins, for example. It can recycle sulfur. It can break down organic material that can be poisonous, for example. And it can sequester things like arsenic that are potentially toxic, or lead or things like that can be potentially toxic. So there are many, many different functions that they have. What's particularly topical at the moment are in relation to climate change. Now, we know climate is changing. Climate cycle, we go through interglacial cycles. It gets warmer and it gets colder. And we're currently in a warming phase. And much of that warming is related to trapping solar radiation in the atmosphere, and it doesn't escape, so we get a bit warmer. Now, what microbes can do is they can influence that process. So, for example, they can fix carbon dioxide in the oceans, and phytoplankton that live in the oceans are responsible for up to 70% of carbon fixation. So they're drawing this carbon dioxide out of the atmosphere and they're suppressing that greenhouse effect. They can also do things like produce and break down methane, and methane is 300 times more effective as a climate active gas than carbon dioxide. And as the Arctic is melting, and the permafrost is melting, methane is being released at a faster rate. So there's a positive feedback loop. If you like, the warmer it gets, the warmer it gets. So microbes can break that methane down and stop it acting as a greenhouse gas. Another thing they can do is they express a protein called an isonucleation protein. And if you didn't have ice nucleators, water wouldn't freeze at zero. It would freeze at something like minus 20 degrees. But what happens with ice nucleators is they allow the ice crystal to form around the nucleating agent. And bacteria can artificially produce ice nucleating proteins on their surfaces to encourage ice crystal formation. And when this happens in the atmosphere, it leads to droplet formation, it leads to cloud formation. And so you can have clouds that are created by microorganisms that then increase what's called the albedo, or the reflectance of the planet. And if you think about, I'm looking at it now, if you look outside the window, if it's a cloudy day, then the ocean is white and reflective and the sun's rays are reflected away again. If it's a clear day, then you can see the darkness of the ocean and all that solar radiation is absorbed. So the more clouds you have, the less energy enters the system. So another thing microbes can do is affect the albedo. They also generate things like dimethyl sulfide, and dimethyl sulfide is a compound it's a bacterial degradation agent of organic matter in predominantly in oceans. So if you go to the beach, the smell of the sea is largely down to DMS. And it's recently been shown that DMS can be produced in terrestrial ecosystems as well. Well, DMS is another very active climate active gas. So another function that microbes can have is entrapment of heat. So there's a wide range of environmental functions. And what we tend to do is pick some key ones to study to understand how the process works. If you want I was just going to say about the papers I've submitted, if you want a very specific example, for a long time, it was always assumed that in the Arctic, the polar regions are all frozen, so that the living things don't do anything there. They're ineffective. They don't have a role. But that was a problem of methodology. And what we're finding now is if you monitor them for long enough in large enough volumes, they do have an action that can be detected. And so one of the things that we've looked at is the decomposition of methyl halides. Now, methyl halides, again, a very climate active gas. Bacteria in snowpack don't appear to influence chloromethane or bromomethane, but we've been able to demonstrate that they do decompose iodomethane. So we now have evidence that bacteria in snow can reduce one of the climate active compounds that can
Yeah.
exacerbate climate change. So getting a handle on these exemplars that we know what
Mm.
bacteria and what their functions are, we can then look back at the community and ask the question, well, how many bacteria can do this function? How
Mm.
often do we find this function present in which different communities? And how vulnerable
Yeah.
is it to being lost from the system?
Yeah. So what's going through my mind, David, is that the irony of Homo sapiens walking around the planet saying we have conquered this is truly ironic, isn't it? I mean, underneath their feet, in their gut, we'll talk a little bit about
Yeah.
the microbiome. Um, there are small things that are actually controlling the whole thing, the whole system. Um, and at some point, they can decide, oh, these guys are completely idiotic, get them out
Yeah.
of here. So that's sort of where we are, isn't it?
Well, two things there. First and foremost, I should stress that most microorganisms, the vast majority of them, 99.9%, are either benign, they have no effect at all, or they're beneficial. We tend to focus on the negative ones because most of the work over the history of scientific investigation has looked at pathogenic species that affect humans. So all of our data is biased and skewed towards the bad ones, but
Yeah,
they are vast minority of the total. So
yeah.
most microbes are good, beneficial, or benign. And then when we say that the beauty of the scientific method is the more we probe and understand things, the more we understand we don't know. So we haven't conquered anything, but we have a better insight into what
Mm.
we're dealing with. And if you think of the human body, it isn't an organism in isolation. It's a community of organisms that live in a synergistic relationship, and
Mm.
without each other we couldn't survive. So we rely on that microbiome, we rely on those microorganisms that live within us to keep
Yeah.
us healthy and keep us functioning. Now, we don't know yet how to manipulate them properly. We have probiotics and we have prebiotics and we have antibiotics, so we can
Mm.
play around with the system a little bit, but
Mm.
it's very naive. It's a bit like an early surgeon poking at a so trying to work out what's going on. What we
Yeah.
do know is we need to understand it much better. So
Mm.
we're very much at the, and I use the analogy freely, tip of the iceberg. We know there's an iceberg there, but we don't know what it contains, how big it is, or what we need to do to it to make it function.
Yeah. So at least we know there's an iceberg there, and that in itself is it's an advancement. Um, so you say here in the conclusion of the paper, our analyses based on ecological networks also showed no clear evidence of recent environmental impact on polar, marine, microbial communities at the
Yeah,
functional level, unless
yeah.
all community sunlights have changed exactly in the same direction and intensity, which is unlikely given we are comparing areas changing at different rates. So if I understand this correctly, David, correct me if I'm wrong. When things change, new organisms step in um, to 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 the gaps, so to speak,
Yeah,
functionally,
yeah,
and in some sense, that is also evolutionary, isn't it? I mean,
very
um,
much so. Yeah.
yeah, yeah. Go ahead. Yeah.
Yeah, so what you do is by removing a function, you create an evolutionary pressure for another organism to be successful in that role. And what the paper concludes is that there's plenty of resilience in the system for
Hmm.
other organisms to have that role, but it is finite. So
Hmm.
you might have, for the sake of argument, 50 organisms with that gene that can all step in one after the other while they slowly become extinct. But
Hmm.
eventually you'll lose all 50 and then the system will collapse because
Hmm.
it can no longer sustain it. But what we've seen from the network observations is there's a lot of inbuilt resilience at the microbial level for
Hmm.
us not to be too concerned at the moment, but it would
Hmm.
be really interesting to know where that limit is, and how far can we push the system
Hmm.
before we start creating a threat that we do need to be seriously worried about.
So
And
mathematically, if I understand this correctly, David, you would say if the trend is, it's not a step function change, it's sort of
Yeah.
trending down or trending up. We have a lot of potential organisms that could step into it. Obviously, when there's a step function change, all bets are off.
Yes,
But we don't know when that's going to happen, right? Is that the way to think
exactly.
about it?
Yeah, and also it may depend on the function. So you could remove nitrogen fixation. It may not be terminal to the community. It might be that they have to shift to some other form of source of nitrogen, but the stability of the system will be shaken because the network system will break down. And I
Mm.
think the important analogy to draw is we're working with microorganisms. And if the same holds for higher organisms, humans might be that one organism or species that's removed from the system. And we don't want
Mm.
that to happen because the system will still
Yes,
function, but we won't be there to do it. So,
yes.
when looking at microbes, we're looking at microbes because we can understand the complexity better. But
Mm.
if it's a universal law that applies to all living things, then it will also apply to higher plants and animals as well.
Mm. So I want to go to another paper, persistent and emerging threats to Arctic biodiversity
Sure.
and ways to overcome them. A horizon scan, you say here. So you say Arctic biodiversity is under threat from both climate-induced environmental change and anthropogenic activity. However, the rapid rate of change and the challenging conditions for studying Arctic environments mean that many research questions must be answered before we can strategically allocate resources for management. Yeah, it's sort of following up from our previous discussion. So there are a lot of unknown questions, and
So
yeah, go ahead. Yeah, go
So
ahead.
you mentioned policy. This paper is really about policy and how science works. So
Yeah,
I'll come back to that. But the
yeah.
important thing is the Arctic is no different to anywhere else.
Mm,
The rate of change of the planet now is unprecedented, and
mm,
evolution needs time to catch up. So if we do things to change things drastically, like removal of the sea ice, then the evolutionary timescale for sea ice microorganisms, for example, to adapt to that change is not the same timeline as the rate of change as it's happening at the moment. And
yeah.
that's why we say with biodiversity loss and with environmental change, it's a threat because
Mm,
we know it's happening much quicker than the environment can sustain and the evolution can keep up with. So
mm,
we might be at that stage where we get this step change, where you get this drastic change in the environment
mm,
that is unrecoverable. This particular paper is a very interesting way about how science works and how it gets funded in the current age.
yeah.
And what happens is that we all know there's a problem. We all agree what the problem is,
Mm,
but how we direct the scientific effort and focus and resources is challenging because I might say the priority is A, somebody in Canada might say the priority is B,
mm,
somebody in India might say the priority is C, and we don't agree, so we all go off in our separate ways. But
mm,
what happens from time to time is the community gets together, as has happened in this particular paper, and the
mm,
initiative was a Canadian one, and a group of people will get together and they'll ask the whole community, what are your thoughts, what are your priorities, and what are the areas we need to focus on?
mm,
And everyone will feed into that discussion and they'll
yeah.
generate a list of priorities, how many there might be, 50, 100, or whatever. And
Mm,
then we'll convene a meeting as scientists and we'll go through and we'll prioritize them and rank them. And
yeah.
if we can agree on the
Yeah.
priorities, we get a paper like this where there'll be 10 to 20 priorities that focuses everybody's minds,
Mm,
focuses the direction of travel, and allows research funders to say, okay, if we put money into this area, it's what the community wants to do. It's what the latest scientific advances need to be. So
mm,
I selected that paper particularly for the policy role of
yeah.
how we formulate policy and how we can work as a group in what would otherwise be a very competitive and um, individually driven world.
And non-optimal. So you say here addressing threats to biodiversity in the Arctic is further complicated by the region's complex geopolitics,
Mm.
as 8 countries claim
Yeah.
jurisdiction over the area with multiple local considerations such as indigenous sovereignty and resource rights. Yeah, so I mean, many of our problems, I would argue, this is my bias, David, is coming the fact that we segmented ourselves to 200 countries.
Yeah. Absolutely.
uh,
Yeah. Yeah. Yeah.
if that didn't happen, we could
Yeah.
solve all these problems actually pretty quickly if you want to. Um, and so there's geopolitics. Um, there are, you say here, we identify research priorities to serve as a starting point for addressing most pressing threats to Arctic biodiversity. Um, from a geopolitics perspective, the question would be who wants to solve the Arctic biodiversity first, Arctic biodiversity laws first?
So, so there's
Uh,
a couple of
yeah,
a few
yeah.
pertinent points here. First and foremost, the comparison with the Antarctic. In the Antarctic, there is no current territorial claims to particular geographical locations. So it's truly an international laboratory for the benefit of humankind.
Mm,
And
mm,
countries work together on scientific problems. There is no exploitation of non-renewable resources. So there's no competing interests in terms of finances and what have you. There's no standing population apart from a few scientists that come and go. So it's a very harmonious place where we can answer some of the questions for the planet as a whole and for humankind in a very ordered and amicable way.
mm,
Flip that to the Arctic, and
mm,
as we lose the sea ice in the Arctic, so the people who have claim to the Arctic have legitimate rights to claim parts of the Arctic as part of their territorial
mm,
integrity and therefore their resources and everything else. However, the challenge is that as you say, the national boundaries are very artificially constructed, and they don't respect things like ocean currents and air masses and
mm,
migration patterns, and that's
mm,
where the challenges come in. And you might say, who stands to benefit? Well, the circulation in the Atlantic, for example, and the
mm,
fisheries therein are driven by warm and cold water currents that are largely driven in the polar regions. So if you mess up the dynamic of the Arctic Ocean, you could potentially affect fisheries very, very far away.
yeah.
You could affect weather systems and weather patterns. You could affect migration of key
Mm,
food organisms like caribou, for example, in North America. So there is always going to be a community somewhere that will suffer from the results of that change. And
mm,
again, it comes down to the time to adapt and the time to evolve. It might be that we can counteract those changes by other activities, but we need
mm,
time to plan and to work out how that might happen. And for that, you really need agreement and consensus and everything else. And we're in a phase now where that is happening. People are trying to build up those communities and those consensuses to try and make informed political decisions. And we as
mm,
scientists can only provide that baseline information to inform those decisions. So they're not driven by commercial interests or national interests, but they're driven by the way the world works and the way the environment functions.
yeah, yeah. So I'm going to go to another paper, David. So microbial metabolism directly affects trace gases in subpolar snowpacks. So you say here concentrations of trace gases trapped in ice are considered to develop uniquely from direct snow atmosphere interactions at the time of contact. So before I go into the details of this, so what do you mean by trace gases?
So, so the key gases in the atmosphere are nitrogen, oxygen, um, carbon dioxide, the ones that we see in the textbooks that we encounter. The common ones that you can buy off the shelf in a laboratory.
Yes.
Trace gases are anything which has a trace amount, so a sub 0.001%,
Okay.
for example, that has a function. It has a chemical function,
Hmm.
and it may have over broad areas might have a significant biogeochemical function, but
Hmm.
it's something that tends to get overlooked because its concentration is quite low.
What are the examples? Do you have a couple of
So,
examples of this?
yeah, so that paper is about as I mentioned earlier, in relation to climate change. They're the methyl halides. So chloromethane, bromomethane, and iodomethane are all
Yeah.
known to be climate active gases that are present in the snowpack.
Hmm.
And because of the difficulty in detecting them at low concentrations and the difficulty of monitoring bacterial activity at low temperatures, it's always been assumed that there's no activity and nothing's happening. But what we know now is what we've been able to demonstrate with this paper
Yeah.
is that the microbes do degrade iodomethane. So over a continental scale,
Hmm.
that activity could be incredibly significant and could
Hmm.
have a major impact in terms of climate modeling. So
Hmm.
that was the first time anyone's been able to actually show that microbes break down a chemical trace gas that has a climate effect.
Oh, wow. Okay. So this is methyl halides.
Yeah.
Um, so you say here our results sort of show large variations of production consumption of other methyl halides. including methyl bromide, methyl chloride, used in climate interpretation. So if I understand you correctly, David, what you're saying is that these are low concentration gases in the environment,
Mhm.
but they have a significant effect in the sort of the long-term environmental effect. And so, for example, if we get rid of the microorganisms that deal with it, um, and they may not be very high in concentration in the microorganism universe, but it could have a significant net effect on the environment. Is that the way to think about it?
Yeah, so if you look at the climate models, the IPCC climate models at the moment, one area where um, they I would say need improvement is probably too strong a term, but
Mm,
where they could do with better parameterization is the role that microorganisms have in the cycling of climate active gases and the role microorganisms have in that process. Because up to this point, they've been assumed to be so low as to be insignificant. But if
mm,
you take the example, if I were to tell you that 1 meter squared of snow has one bacterium in it, it's insignificant. It's not going to have any difference. It's not going to make any difference to anything.
mm,
And even if you look at the cells over which climate models are based, the number of bacteria probably aren't significant. But if you
mm.
multiply that 1 meter squared by the size of the Antarctic and
Yeah.
add then the size of the Arctic and all the cryospheric areas and all the
Mm,
atmosphere that's less than 5 degrees and all of the ocean that's less than 5 degrees, that's a very, very large number of bacteria. And
mm,
even if the turnover rate is very slow, globally it becomes a significant number. And we also
mm,
have to think about the time periods involved. So if we think about 1 year, that bacterium is not doing very much. We can forget
mm,
about it. And that's legitimate because it's doing so little. To all intents and purposes, nothing's happening. But if you take that area of bacteria over a glacial cycle, that's
mm.
an awful lot of transformation that's happening that could have an incredibly significant effect. And
Yeah.
it's only recently that we've had the technological capability to demonstrate it. So we
Mm,
might have hypothesized that it was happening, but now we've been able to show that it's happening. Now we've shown it's happening, we can
mm,
start to work out what those flux rates might be and how common those flux rates are so that
mm.
we can feed that data into the next generation climate models.
Yeah, yeah. So I want to go to another one, David. So aerobiology over the Southern Ocean. So let's go to Antarctica. Implications for bacterial colonization of Antarctica. So you say here parts of the Antarctic are experiencing dramatic ecosystem change due to rapid and record warming, which may weaken biogeographic boundaries and modify dispersal barriers, increasing the risk of biological invasions. Um, in this study, we collected air samples from 100 locations around the Southern Ocean to analyze bacterial biodiversity in a circumpolar air around the Antarctic continent, as understanding dispersal processes is paramount to assessing the risk of microbiological invasion. So it's not just Antarctica, the continent; it's the environment around it, isn't it? I mean,
Yeah.
that's a complex system. Yeah.
Well, so we've been talking about environmental function. Now, one of the theories used to be that because bacteria are so small, everything is everywhere and the
Yeah.
environment selects. So there was a view that bacteria are so numerous and so small that every species is represented everywhere and that
Mm,
it's just chance whether you find it here, there, or where. What we're realizing now, because of the higher resolution of our techniques, is that everything is not everywhere. And you
mm,
do have biogeography of bacteria, and certain species occur in certain regions of the world and they don't occur everywhere. So our area of interest is the Antarctic and the Arctic because it's changing quite rapidly. It's warming in particular 3 to 4 times faster than other parts of the planet. So
mm,
we can go to the rapidly warming parts and see what's changing and how it's changing, and
mm,
then extrapolate that to what would happen if this happened globally. So we can go to the areas where, for example, catastrophic change happen. If a glacier melts, then you're changing from ice to soil.
yeah.
And that's a fairly catastrophic change. But we can
Yeah.
study the microbes and what the functional differences are between those microbes in the ice and those in the soil. And we can start to make predictions about what might be happening. Now,
Mm,
for functions to be changed, you either need to lose functions or gain functions. Now, we've predominantly been talking about losing functions, but
mm,
there's also the gaining functions. that
Yeah.
is associated with migration of bacteria from one place to another place.
Mm.
Now, what that study showed, or what we found in that study, is that the Antarctic circumpolar current, or the polar front, acts as a selective dispersal filter. So
Yeah.
not all bacteria can travel into and out of the Antarctic. Some of them can, some of them can't.
Mm.
But what we're seeing under a changing climate scenario is that that barrier is weakening. So we
Mm.
may end up in a situation where all bacteria can move freely across the polar front, and we get colonization of Antarctic environments with bacteria that would otherwise have not got here. And that's exactly the project I'm working on now. So
Mm.
I'm in the Arctic investigating whether that same scenario also
Mm.
applies to the Arctic Ocean, because the Antarctic is a continent surrounded by sea, whereas the Arctic is a shallow sea surrounded by continents.
Yeah.
But we
Yeah.
still have these stable air masses on top of them, and
Mm.
we don't know how well microbes can move across them. What we do know in the north is
Yeah.
that the jet stream is starting to destabilize, and we're getting inflow of microbes much further north than they used to come. So the
Mm.
question then is, can they colonize, and can they change the functional potential of
Mm.
the recipient communities?
Do we know enough, David, to sort of artificially intervene? What I mean is that, can we seed um,
So,
these
yeah,
areas? Yeah.
it's challenging, but it's not impossible. I mean, all things are possible given enough time and resources. So, one of the movements at the moment, which is controversial, is called geoengineering. It's
Yeah.
a way of changing the environment to address the balance of our insults. So, for
Mm.
example, bioremediation might be one example, biosequestration might be an example. But what we're seeing proposed now are things like spraying bacteria into the atmosphere that have
Mm.
isonucleating genes on them, so that you're artificially creating clouds that can reflect the sun's light and reduce the temperature. Now, small experiments have been done. Another experiment has been done in the Southern Ocean. We know the Southern Ocean absorbs most of the atmospheric CO2 that gets absorbed by the oceans, or a large
Mm.
proportion of it, and it's iron limited. So if you add iron to the ocean, it can absorb more carbon dioxide. And so there have been experiments done where ships have traveled through the Southern Ocean, dropping lots of iron in the
Mm.
hope of drawing down more carbon. And the jury's still out on how effective that is, because some of
Mm.
that gets recycled and emitted again. Some of it ends up being sequestered into deep sea carbon. But it is an example of what we can do to change the system to our benefit, or to manage the system to our benefit. The
Mm.
biggest challenge is in almost all conservation situations where humans have tried to intervene in natural processes, we've tend to create unforeseen side effects we haven't been able to predict before.
Yes.
So,
Yes.
so the risk is always, in doing that, do we cause that problem, that problem, that problem?
Yeah.
Or
Yeah.
do we just address the main feature? But until we understand the systems, we're not going to be able to come up with policies that can help or address those challenges.
Yes, I fully agree with that, David. Um, yeah, every intervention that we have done resulted in disaster. I was heading more toward, if you understand the species that is doing the work,
Mm.
and for whatever reason it is disappearing or reducing, can we repopulate the species? I mean, any sort of engineering intervention has been complete disaster for humanity forever. But this
Well,
could be some sort of a biological intervention. Yeah. Go
I
ahead.
wouldn't say that, definitively, because the ozone hole is
Yeah.
a good example of a good news story where we've discovered a problem and we've done something about it and it's resolved itself. So it's not a hard and fast rule. We always mess it up, but normally there are unforeseen circumstances we haven't predicted that we
Mm,
call. There's always, it takes time to understand properly the system we're working with.
that's
Now,
true.
the
Yeah.
key thing, or the current technology, is genetic modification. We
Mm,
can take a common bacterium like Pelagibacter in the sea, and we can insert a methane degradation gene, for example.
yeah.
And if we release that and it's able to compete with the local population, then it's effectively introducing a function into the environment. But
Yeah.
at the moment, that's very unpalatable. We're not,
Mm,
as a community, we're not happy about engineering microbes and releasing them to the environment,
yeah.
because we don't fully understand
The
that
side
microbes swap
effects.
genes
Side
around.
effects are not
Exactly.
predictable. Yeah.
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 Microbes are promiscuous. They switch genes around. They swap things around. We don't understand the processes that control that. So we
Mm,
might put in a methane degradation gene that could end up being inserted into a iron sequestration gene and mess up iron sequestration. So, we can do the experiments in the lab and we can do theoretical experiments and we make predictions about what we'd like to do,
Yeah.
but it's very risky to do it in the environment. One thing, there's a couple of examples of things we can do, and that's trapping radionucleotides in the environment. Deinococcus radiodurans is a very radiation tolerant bacterium. And if you grow it in a solution of radionucleotides, it can absorb those radionucleotides and then you can filter it out of solution. So it's removing soluble radioactive present in drinking water. Well, sorry, I've internet connection problem there.
No, you're
Campylobacter's
okay. Yeah.
one way of removing arsenic from solution. So we can do those in bioreactors in factories where we put the water from the environment through the bioreactor and we return it and we don't put any bacteria back with it. So we can have that kind of impact, but that tends to be on a much smaller scale. When we're talking about engineering the environment, I think we're some way from that yet, but people are interested in doing it, and there are proposals coming in where suggestions are being made.
Yeah, so I want to finish up, David. I know that you have a dinner coming up here. The crewed journey to Mars and its implications for the human microbiome. I have very interested in this. So you say here a human space flight to Mars is scheduled for the next decade. In preparation for this unmatched endeavor, a plethora of challenges must be faced prior to the actual journey to Mars. Mission success will depend on the health of its crew and its working capacity. Hence, the journey of Mars will also depend on the microbiome, its far-reaching effects on individual crew health, the spaceship integrity, and food supply. So I can quite remember, David, the number of cells in a human body is it like 10% of the the total number of cells or something like that?
So there was speculation at one time there were 10 times the number of microbial cells in a human body than there were human cells, but that's been challenged more recently, and it's more of a range. But it's of the order of magnitude of at least the same number of cells in a human body, if not more. So there is drastically more than we thought there were.
Yeah. And so microbiome. So I mean, we have two problems. We don't put the spacesuit on this microbes when they go to space. So they're sort of swimming around on their own. And I would speculate that they will change their behavior in no gravity situation. And so the microbiome is going to change regardless, right?
So scientifically speaking, we wouldn't dream of sending humans to Mars until we properly understood the environment, because one of the first things we would want to do is look for the presence of life or the history of life on the planet before we sent humans there. But the trouble is, unless we send humans there, we haven't got the human drive to explore behind it that provides the financing to do the exploration. So we're, as scientists, caught in this conundrum that we have to do it in order to make it happen, but it's not the best scientific practice. So what we have to do is try and understand it as we're doing it. And one of the things we know is going to be affected is the microbiome associated with the astronauts and the astronaut health. If it takes them 3 years to get there and come back, if something happens to them on the space flight where they become ill, how do we know they're ill? How can we treat them so they're no longer ill? And how can we know if something's happened to them that's detrimental to both their health and other humans that may come into contact with them? So say, for example, a microbe was to become more pathogenic in their microbiome as a result of being on that space journey. And then they come back to Earth and we get them out of the spacecraft, and all of a sudden this new organism causes a problem. We need a way of understanding that before we subject everybody else to risk. So that paper really was looking at proposing and looking at the human microbiome, monitoring the microbiome for what we call a state of dysbiosis. And dysbiosis is when the steady state's broken in some way. to understand whether there's something we might need to look at or understand. And it was talking about how often we might need to sample astronauts, what we would be looking for, how we might remedy it, because we can do those experiments on the space station, and we can do those experiments on the Mars bases that become the forerunner of the Mars
Yeah.
expeditions. So looking at the microbiome in astronauts is quite a good way of understanding their fitness and health. And not to mention the planetary protection issues of when the astronauts get there, because spacesuits vent. So you put an astronaut onto a planet and they vent the pressure, and then they're
Yeah.
releasing organisms onto the planet, albeit they probably won't survive. But if our hypothesis is there are no organisms there, and we go looking for some, and we find some that we've taken there ourselves, it
Yeah.
completely undermines any and the scientific integrity of the endeavor.
So what data do we have from the space station? I know that, a couple of people got stuck there recently.
Yeah, so
Um,
there's really good
yeah,
data from the space
yeah.
station. The astronauts have had their microbiomes monitored on the
Mm,
space station, and there have been regular swabs of the space station itself to look at the accumulation of different microorganisms in the space station environment. So there's a lot of data starting to be collected,
mm,
but we're still right at the limits of the resolution of what can be detected and understanding what impact that resolution will have
mm.
for the astronauts and their health.
Yeah, so I had to get so it's a complex equation too. It's not just sort of looking at the change in the microbiome, but rather predicting what the change is going to do. Some of the change could be beneficial,
Oh yeah,
potentially.
yeah.
Yeah, yeah,
Oh, going back, if we draw the analogy with the environmental sample and the
yeah.
networks and the stability, the microbiome, it tends to be stable. So what we're looking for is a change in that stability, because the change in stability will suggest there's a problem, whether it be
Mm,
positive or negative. It should not destabilize the microbiome. So if the
mm,
microbiome becomes destable, then it's like an alarm bell going off. We know that there's a challenge that we need to investigate. But if the microbiome stays stable, then we know that the individual is probably likely to be healthy.
mm. Excellent, David. So I know that you have to go and have a dinner. What's the temperature outside right now
It's not too
in the
bad. It's the
Arctic?
summer here, so it's round about. It drops between 0 and -10, so it's quite. Some of the snow can melt. It's
Yeah,
not particularly cold. We tend to do the field work in the summertime, so in the Arctic it's this time of year. In the Antarctic it tends to be over the winter, but
yeah.
no, it's not too
It's
bad.
like Chicago or Minneapolis in the winter. I mean, about
I
the
always
same.
remember my first season in the Antarctic. I rang my aunt who lives in Toronto in Canada,
Yes.
and she said, "Oh, what temperature is it where you are?" So it was -25, and she just laughed and she said, "It's -40 in Toronto." I said, "Yeah, maybe I didn't need to come too far."
Yes. Yeah, yeah. So the Arctic is much warmer than the rest of the world. Um,
A common misconception about the polar regions is it's not the fact they're cold that's the challenging part. It's a
mm.
polar desert because all of the water is ice. It's
It's
not
very
available to
dry.
organisms. So it's
No,
very dry,
no humidity. Yeah,
low nutrients, no
yeah,
population. There's very specific features of polar regions that make them different from other parts of the globe. But the cold is just an incidental.
mm,
It slows things down. But when things are frozen, they're frozen. When they're cold, they carry on doing what they would do, but they do it much more slowly.
yeah. Excellent, David. Yeah, thanks so much for spending time with me from a very inconvenient position. uh,
It's been a pleasure, and I extend the invitation to any of your guests or listeners or watchers. If you want to chat or have any questions, feel free to email me, and I'm more than happy to talk about science and engage with the wider community about what we do and why we're doing it.
yeah, absolutely. Yeah. Thank you. Thanks, David.
Pleasure. Bye for now.